guest, who is the Pro-Vice-Chancellor of the Faculty of Health and Life Sciences, Professor Louise Kenny, who's going to actually introduce Bernadette to us. I think Louise has known Bernadette longer than I have, which is still some time. And so I'm going to hand over straight away to Bernadette. And of course, I want to welcome, to Louise. I want to welcome Bernadette and all her family, but I think Louise is going to do that especially. So well done for making it. I know this is scary, Bernadette, but you're amongst friends. Over to you, Louise.
Thanks, Pat. So, yeah, it's a great pleasure, huge pleasure and honour to welcome all our guests this evening to Bernadette's inaugural lecture, and also to this fantastic venue, the LMI, which is, many people believe, the spiritual home of medicine in Liverpool, which is therefore quite a fitting place for the lecture that we're about to receive. As Pat says, I've known Bernadette quite a while now, and I was trying to think as I was walking over here this evening when we first met, but it was in Ireland, I think. And those of you who know Bernadette will know that she's from Ireland, and Bernadette reached out when I was working in Ireland, actually on science, but also on women's rights issues, I think is one way of politely describing it. And her support, Bernadette's support of what I was trying to achieve in Ireland at the time, made a huge difference to me at a really difficult time in Irish politics. And yeah, so it is a fantastic honour to be giving the introduction to her inaugural tonight. And I'm really thrilled that when I came back to Liverpool, we managed to entice Bernadette to join us from Leeds, to move down the M62 and join us in our endeavours here in Liverpool. I think inaugural lectures I remember my own inaugural lecture. It is one of the most awe-inspiring lectures you can give. It's also one of the most difficult because the audience comprises of your harshest critics, your family, and in my case, my kids, one of whom fell asleep on the front row. He'd been on call the night before. That was his defence. And also experts in your field, people that you work with and publish with, who know your work really well and sometimes better than you, as well as people who just are interested in what you're bringing to the university. So it's a really diverse audience. But as Pat says, it's one of the most friendly. Everyone's here because they really dig you, Bernadette. So please do enjoy us. I know we're going to have a fantastic lecture on a topic close to Bernadette's heart, which she's devoted most of her career to, and it's the molecular mechanisms underpinning the role of nutrition and lifestyle in liver disease. When Bernadette joined us in September 2024 as chair in nutritional biochemistry, it was as our first real chair in nutritional biochemistry to do something we think quite innovative at the interface of where nutrition hits pharmaceutics and hits public health and hits biochemistry. And I can think of no one better to do that, given Bernadette's long background and significant achievements in uncovering the molecular mechanisms underpinning diet in health and disease. Many of you will know that Bernadette, prior to coming to Liverpool, was at Leeds, and prior to that, she enjoyed a really illustrious start to her career, where she gained her PhD at the University of Florida. She was awarded many prestigious prizes as an undergraduate and postdoc. And really, that was the origin of what has been a stellar rise to the top, firstly in Florida and then in Maryland, before moving back to her home in Maynooth in Ireland. She's been in the UK with us since 2008 and has had a fantastic contribution to the universities where she's worked, and that's been recognised by many external prizes, including in 2018 the Nutrition Society Silver Medal for Research Excellence. In this lecture, Bernadette is going to share with us her journey on the road less travelled to professor and chair, and her insights into what is a really contentious, often contentious subject, and that's nutrition and how we can eat to prevent cardiometabolic disease. And given recent NHS sort of statements on this area and the leftward shift into prevention, this is incredibly well timed, as well as being of huge importance to public health. So I'm going to sit down. I know we're all in for a treat for the next hour, and I want to ask everyone to give Bernadette the warmest of Liverpool welcomes to the podium.
Thank you very much, Louise, and thank you to everyone who's here and online. If anything, I'm a little overwhelmed, but I promised myself and my dear friend Malcolm that I won't get emotional until later this evening. And that's partly because, as many of you know, I do like to tell a story. So what I've done for myself to keep emotions at bay is set myself some objectives. And the first of those is that I do want to share with you what I believe has been quite a circuitous and unusual road less traveled and sort of scientific and personal journey. And mostly, if anything, I want to honor my mentors, my students, my collaborators, the people who've kept me on that road when it's gotten treacherous and difficult and painful. But separately, as a lecturer and a teacher, I can't help but want to share some of my knowledge. So I aim today to share with you a little bit of the complexity and the beauty of molecular mechanisms that underpin the roles that dietary nutrients play in health and the lack of dietary nutrients in disease. I want to explain a little bit about systems biology, especially to those of you who might not know what that means, and talk more about systems thinking and systems analyses and how these can help us understand what I will explain as the emergent phenotypes of obesity and steatotic liver disease. I'm going to talk a little bit about my current and the future work that I plan here in Liverpool, and I'd like to end with big picture. And I can't give you all the details of all of the evidence, but as Louise alluded to, we have never lived in a noisier, more contentious space. I struggle to compete with podcasts and the longevity bros, the misinformation, the disinformation. So I'm going to distill down my takeaways on nutrition for healthy aging. So I'm focused on the prevention of steatosis or fat in the liver, but equally these messages apply to type 2 diabetes, cardiovascular disease, hypertension. They're all quite similar. So my road less traveled began when I was 12, and when we moved from the very small island of Ireland to the very large continent of America, and we moved to Florida, and this was kind of defining. And unlike some of my colleagues, I did not show an early penchant for science. I did not do A levels in biology and chemistry and the like. But reflecting back, and this has been easier because we're going through photo albums as we plan our move, what I can say in high school is that I was an avid reader and a creative writer. And actually, I won several awards for essays that spoke to my identity as an immigrant, and very poignantly in the context of current political movements, my belief in the US Constitution and what that meant. Now, it was the 1980s in Florida. Top Gun was a big thing. We were very close to Cape Canaveral. I had a really smart best friend who I was easily influenced by. And I had grand aspirations when I set out for university to study aerospace and aeronautical engineering. Needless to say, speed bumps were met along the way, particularly when I decided, although I did really love differential equations and I loved graph theory, the mathematics of the threads on screws and bolts were less interesting to me. So I had a change of heart midway through my university, and driven mostly out of my passion at the time for exercise and nutrition, I switched to nutrition. Now, ever one to change her mind, by the time I had finished my degree, I decided that I never wanted to tell people what to eat for a living, that people mostly eat, what they learned to eat as children. And instead, I was in management and retail management for a while. However, I quickly got bored again. And a few years later was questioning whether or not I wanted to go to medical school. And in going back to the department that I had gotten my undergraduate degree, I had the great fortune of meeting this woman, Dr. Kathy Levinson. And Kathy was new on the faculty at the time, but it is her who exposed me to sort of the magic of molecular biology. And I started working in Kathy's lab, learning how to isolate RNA. Kathy could make everything seem like fun. And it was with Kathy that I published my first paper, which was related to changes in gene expression in the brain in response to exercise. And to me, this idea, this notion that what we eat, what we do every day, that how this works is by altering these molecules at the cellular level, was all just pretty amazing. The other thing that Kathy did, and she said at the time with some nerves, is she introduced me to one of her mentors, Professor Bob Cousins. He was then and has only continued to be an eminent scientist in the field of zinc biology. And so what I'm going to do, particularly for the people who are not molecular experts, is show this one small schematic which shows you how genes are regulated in response to dietary zinc. Dietary zinc is an essential mineral. Dietary zinc deficiency remains one of the most prevalent micronutrient deficiencies worldwide. And what this schematic shows is that there are very specific proteins, intracellular proteins, here labeled MTF1, that bind zinc and in response to that move into the nucleus and activate a host of genes. These include genes that then bind zinc, like metallothionein here in MT, and they include the molecules that transport nutrients in and out of cells. And at the time when I joined Bob's lab, although we knew bits of these stories, Bob was keen to characterize, and at that time, most of the transporters had not been identified. So I joined Bob's lab in 1997, and Bob ran a tight-knit group, and he really believed in the diversity that is required in terms of science. And so in this picture here, there's actually 5 countries represented and a group of PhD students, postdocs, and visiting fellows. In particular, I worked very closely in the second picture with Dr. Ray Blanchard, who had established what was state of the art at the time in the late 90s for identifying genes that were regulated by whatever differential display. Now, Ray was working in the intestine because one of the symptoms of dietary zinc deficiency is diarrhea. And the other symptoms of dietary zinc deficiency, particularly in children, is it's an immunodeficiency. The thymus involutes, it gets smaller, it shrinks. CD4, CD8 cells, they don't mature the way they're supposed to. And so I decided in joining Bob's lab that the thymus was the tissue that I would work in. And I was also interested in developing an animal model that wasn't of the very severe zinc deficiency that we knew about, but what happens earlier than that? What happens before the T cells change? So this early work, and I'm not sure if I know how to use the pointer here. Here we go. Hopefully everybody can see that. My early work identified that in response to the loss of zinc, this particular intracellular signaling protein, lymphocyte-specific tyrosine kinase, was upregulated, presumably as a method to try to reestablish that signaling. And this work was published two years before the solution structure of what is now known as the zinc clasp. So zinc is absolutely essential for all downstream signaling of the CD4 and the CD8 T cell receptors. It was a heady time in the lab at the turn of the century. I love saying that, the turn of the century. It seems so long ago. And overnight, you know, the ease of identifying genes changed when the human genome sequence was published at the turn of the century. So it was a really productive time during my PhD, and Bob's lab systematically characterized the immune response and the response in the intestine to changes in dietary zinc. This is me at my PhD graduation, and this is what I refer to as Bob's happy face. And he's been an incredible mentor to me. He's online now, and I'm absolutely thrilled. In August of this year, he's been awarded the Living Legend Award by the International Union of Nutritional Scientists, and I am looking forward to seeing him in Paris. Now, it was a busy time. And so by the end of my PhD, I was already questioning what I wanted to do with my life again. And so what I did was I actually unusually took a science policy fellowship. And this was quite, I don't think I even really appreciated how prestigious it was at the time, but this brought me to the Institute of Medicine and the National Academies of Sciences. And they had just opened a new building, the Keck Center, which had this amazing DNA staircase that I used to walk up and down to move from my desk. And for the biochemists in the crowd, this is Bruce Alberts, who was then the president of the National Academy of Sciences. But what I worked on, and I'm privileged to be named even on this forefront of this matter, was about stakeholder engagement and how government works and how we pull together huge reports. And this was a really seminal report at the time on what we should be doing to prevent childhood obesity. Many of the recommendations in that 300-page report have yet to be implemented. But Bob was right. He predicted that I would find things slow going. And I did realize that I didn't like being behind a desk. And so I went and did a postdoc at the NIH and had the formidable experience of presenting data to two NIH directors at the time. Francis Collins went on to be the director of the entire NIH. If you were to look at my CV, I didn't make a huge mark on the multiple endocrine neoplasia field at the time, but I did learn a lot. And in particular, I embraced proteomics, having done transcriptomics in Bob's lab. But I found the environment different. Bob ran a team environment. The NIH was extremely competitive. And one of the things that I did to stay sane was I went to every single lecture that was ever going, any Nobel Prize winner, Sloan Kettering Mott, whatever the prize winner was, preferably if they were really senior and telling their battle stories, I went to these lectures. And one of the lectures that I went to that was really pivotal in terms of changing my thinking was Leroy Hood. And for those of you who don't know, this is a really remarkable man, considered one of the founders of systems biology, and one of a very small number of people who have been admitted to all three national academies in the United States. And what he said in that lecture that captured me more than anything was that nutrition, along with plant biology, was really an area that could really benefit from systems biology and systems approaches. So I'm going to do my best. I've got Doug in the crowd, so that is really nerve-wracking to explain systems biology. But essentially, this is founded in mathematical graph theory and cybernetics. And it's a way of deconstructing anything into a network. And so when we're thinking about humans, you can deconstruct them into genetic, molecular, cellular, organ level networks. And we can also, and this is really important, visualize humans as being part of social networks. And this can really explain things in terms of the context of environmental and economic food environments, etc. So for the last couple of decades, systems biology and systems medicine has attempted to get to a place, and work is still ongoing, where we integrate a variety of data sets, clinical, genetic, multi-omics, to what we call an in silico human. And the ultimate goal is to predict what we call emergent properties. So to kind of give an easy explanation of an emergent property, this is sort of, a whole is greater than the sum of its parts. So if you think about a car, if I showed you a bunch of car parts, especially to the uninitiated, it would be difficult to envision the emergent property of transport. So in systems pharmacology, Ideally, what we're trying to predict is who's going to respond to what drug, who's going to respond to what dietary nutrient, what are the off-target effects, and can we predict these? So network approaches and network biology has been used in a variety of different systems. This is a visualization of a food system, an urban food system, which involves producers, retailers, fast food outlets. Systems, social network analysis and systems theory can also be used to understand information flow or misinformation flow or disinformation flow on social media. And this is a work of a recent PhD graduate, Dr. Ellis, with myself and Charlotte. And one of my favorite examples of where systems have been used to explain a very complex phenotype is this piece of work, which was also quite seminal because it was published in 2007, right as I was beginning to start my first laboratory. So it's okay that you can't see every detail on what we lovingly call this plate of spaghetti. But in the center is energy balance, and the committee that put together this map, and it is a qualitative map, was trying to explain the complexity of variables and determinants in obesity. And what their work shows, and it's frustrating that people still haven't fully understood this, is that it's not one single thing. It is what emerges from the totality. At the center of energy balance is really dictated by a combination of biology. So it's not just genetics. Genetics are important, but behavior is essential. And we've got a really great appetite and obesity group here at Liverpool, physical activity and food intake. And you can't see all of the things that determine this, but just to point out, physical activity, and when I tell you at the end, you know, what I know about nutrition, I do so with the humility that many people can't act on our advice. So it's okay to tell children that they need to exercise more, but if they live in a very unsafe urban environment, that physical activity might be difficult to get to. So parking this back for the moment, I'm still in DC. I'm not completely happy in my lab environment, but I am doing my best to network. And this is a picture of the Irish Embassy, where I had the good fortune to attend a networking event for Irish American scientists. And at that event, I learned about the Marie Curie Fellowship Scheme. And in wanting to leave the NIH as fast as I could, I went looking for Marie Curie fellowship opportunities. And I found one in Maynooth University. And for those of you who don't know, this couldn't have been more of an antithesis environment to the NIH. It was originally a seminary. So there's this old, beautiful side of the university campus. But I was joining a very brand new custom-built Institute for Immunology and the lab of Dr. Shirley O'Dea. So again, in terms of a journey that isn't completely predictable, I can't say that I left a huge mark on epithelial lung biology, which was Shirley's subject, but I learned a whole bunch of things during my 2 years there. The first is that Shirley, and this is quite personal, but Shirley normalized for me that female scientists could have babies and they could take 6 months maternity leave, because coming from America, that was not true at all. I also learned that I absolutely loved academic science, and I loved the combination of teaching and doing research on a, I suppose, smaller scale than the NIH. Other important things happened. I met a fast-talking Frenchman, much to the disappointment of my Irish family, who thought I'd settle down with a nice Irishman. And I made friends, lifelong friends, with an amazing group of women. And we're pictured here. The reason people have Guinnesses in their hand is we were hosting the British Association for Lung Research, and we had the conference dinner at the Guinness factory, which was quite exciting. Whatever was in the water in Maynooth at the time, we've stayed friends. And in this picture are represented 3 professors, 3 senior scientists in industry, and 1 entrepreneur. And there's other people who came of age at the same time who were also really successful scientists. So a small university, but packing quite a powerful punch in terms of its legacy. So it was clear lung biology was not going to be my cup of tea. And at some point, Shirley memorably said to me, "What do you most want to do, Bernadette?" And my answer at that time was a bit vague. And it was that I knew that I needed to have a research question, which was grounded in nutrition, that nutrition was my passion. So what I did, which is what I have done throughout my scientific career, and it's always stood me in good stead, is I went to the literature. And identified at that time a disease that was then known as non-alcoholic fatty liver disease. And very simplistically at the time, it was essentially a disease that was named by elimination. So when hepatologists didn't have any other reason for fat being in the liver, it was labeled as non-alcoholic fatty liver disease. And this is an umbrella term, and it encompasses steatosis, which means fat in the liver, steatohepatitis, which means fat plus inflammation, and severe fibrosis and cirrhosis. So back in 2007, there was no markers, there was no therapeutics. I viewed this as being quite understudied. It was very much under the radar, certainly in the nutritional sciences. We didn't know about it. And for me, it was closely associated with obesity and metabolic syndrome. So I felt like that was an area that I could make a mark. And interestingly, in my first review paper on this subject, which I wrote while I was on maternity leave, I sort of grandly titled it The Hepatic Consequence of Obesity and Metabolic Syndrome. And this has turned out to be sort of prescient, because in 2023, after 2 years of a global Delphi consensus round, this disease has been renamed. And it's been renamed based on its phenotype. So the phenotype is fat in the liver, and that fat in the liver can come from multiple causes. So what I spend most of my time focusing on is what is now called metabolic dysfunction associated steatotic liver disease. This is a lot of alphabet soup, or MASLD. And the change in nomenclature is really significant, because what it does is it changes non-alcoholic fatty liver disease, which was a disease of exclusion, to a disease that is positively diagnosed based on the presence of fat in the liver and abnormal metabolic parameters. And that's distinguished from alcoholic associated fatty liver disease. The other really important change that has come with the change in nomenclature is that there's this new mixed category, so MET-ALD. And this is, again, something that I've recognized for some time. So many people who are living with obesity also consume more alcohol than they probably should. And it might not be at the level of what causes alcohol-related liver disease, but it's definitely somewhere in the middle. And we don't have a good handle on what the linear range is. So this has opened up a lot of research. And this diagram illustrates that this disease is in fact progressive, and there are other causes that can cause steatotic liver disease. And this sort of nomenclature, moving to naming a disease by its phenotype, is not unusual. Within the hepatology field, hemochromatosis has similarly been renamed, as well as primary biliary cholangitis. So this schematic attempts to distill a lot of our understanding on steatotic liver disease pathogenesis. I know that there's a mix of expertise here, but in brief, fatty acids in the liver can come from dietary sources and alcohol. So sugars in the diet, including sugars from alcohol, are converted by a process of de novo lipogenesis. Fats in the diet circulate around through lymph, and they can end up at the liver. And importantly, adipose tissue, the release of non-esterified fatty acids, is a primary source of fat coming into the liver. And under normal circumstances, that's really important. So when you're fasting and your blood glucose is dropping, you want to release fat from your adipose tissue to make glucose in the liver so your brain stays well. And in brief, these fatty acids are oxidized for energy. In that process, they generate reactive oxygen species. The dark lines here are normal physiology. So this is normal. You're breaking down fatty acids. The same long-chain fatty acids can be built up to lipid intermediates, triacylglyceride. And that is, again, all a normal part of metabolism. But the difficulty is when metabolism is overloaded. And what we've really learned over the last 20 years is a couple of things. The first is that these lipid intermediates are not just intermediates on a metabolic diagram. They're signaling molecules in and of their own rights that determine insulin resistance and other things. And it's the flux through the system. So if you have an excess of lipids, they're put into lipid droplets for safety, really, but this can become pathological. Triacylglyceride should be exported into the circulatory system. And again, this can be normal, it can be pathological. So the other thing this diagram tries to point out is that the gut microbiome plays a huge important role. Adipose tissue, 20 years ago, we thought this was just a sink, but we now know it's an important source of inflammatory molecules. And in the background are meant to be strands of DNA. I don't know if you can recognize that. But when I started, there weren't any genetic polymorphisms identified for this disease, but there are now. So my overall take on this is that steatotic liver disease is an emergent phenotype. And I just briefly want to mention, throughout my career, I've tried to focus on children, partly because early on I recognized that children were kind of a good model of the disease. They're a different model, but usually alcohol isn't a confounding factor. And in children, things are a bit different. So the onset of steatotic liver disease in children can really be driven by early life factors and growth patterns. So pre and postnatally, particularly if they're born from a woman who's living with obesity, gestational diabetes, diet, so saturated fat, which I'm going to come back to a few times in this talk. High saturated fat diets in pregnancy can lead to steatosis in newborns. And then other factors like whether or not a baby is born by cesarean, or a normal birth, whether or not they breastfeed or formula feed, how quickly they grow. All of these impacts will affect both the gut microbiome and their propensity to move to progressive steatotic liver disease. So going back to the question, what do you most want to do, Bernadette? In addition to going to the literature, I went to my first Nutritional Society conference. So this was in 2007 in Coleraine. And I had the very good fortune of being introduced to this woman, Professor Margaret Raymond, who said, oh, well, we're hiring a lecturer. And I didn't admit to her at the time that I had never heard of the University of Surrey, but I quickly looked it up. Unbeknownst to me, thanks to Bob's papers, I had never heard of the ref, but I was still very competitive. So I did have other job offers at the time. But Margaret went to bat recruiting Fiona Green, an eminent geneticist from Oxford who had moved to Surrey, and Linda Morgan, who's a peptide gut hormone expert, and Sue Lanham-New. So it was first Linda and then Sue, who were my two heads of school. And Sue is an international expert in vitamin D, so you'll see where that inspiration came from. One of the things that Shirley had taught me about was research planning. And so I started the lab with really clear questions, kind of big ones, but very clear in my mind. And that was, what are the dietary, genetic, and molecular factors that influence pathogenesis and progressive disease of what was then called NAFLD? Could we identify molecular markers? At the time, there was no biomarkers. And could we model the response to hepatic lipid loading in a systems fashion? And then how best to intervene with nutrition and lifestyle. So 2008 to 2015 was a very productive time, not least because I decided to marry the talkative French guy. And in 2009 and 2011, first Claire and then Xavier were born. And, you know, there was a lag time in terms of publications, but science got done nonetheless. And perhaps a key to my success was recruiting my little helpers. And this is Claire filling pipette tip boxes and Claire organizing my marking. And this is Xavier. This was at the European Proteomics Conference in 2012. And it was through these proteomics conferences that I met Doug and I met Claire and I met Rob Bain. And the other thing I want to say, especially to any young aspiring female scientists or the men who are going to support them, is that the reason Xavier was there is because I was still breastfeeding, and I had gone to Sue very stressed because the university budget for a hotel room wouldn't cover me bringing Nick and the two kids. So anyway, and Xavier loved the lab early on. Okay, so those of you with a sensitive health and safety disposition, look away. So it was a productive time, and this is Christos Spanos, my very first PhD student, and he embraced the challenge of trying to do discovery-based proteomics, first working in animal models, and then trying to translate to human steatotic liver disease. And we did indeed find an enzyme, glyoxylase, which was dysregulated in, these are biopsies from children with fatty liver disease. And then in terms of non-invasive biomarkers, the substrate of that enzyme you can see increasing with BMI in adult patients. And I won't give you all the details, but we identified the mechanisms by which this dysregulation happened. Now that's a nice, pretty story, but I just do want to point out and address the lag time and my long road to professor and chair. So Christos started early on and the paper got published in 2018. And Doug told me the other evening that I have to tell the painful stories as well. I won't give you all the details, but this was a labor of love involving multiple institutions, multiple clinicians. There was a battle to get it out the door the first time, and we eventually got it published. And I think it was, I know it was accepted on Valentine's Day 2018. And what was interesting about this particular painful story is in 2019, our results were confirmed by a really huge analysis, a systems analysis that linked quantitative trait loci to proteomics. And they cited us. They had submitted to Nature 2 weeks after we published, and I was asked to write commentary in that lesser known Nature journal here, which I did, but nonetheless, it was a point of vindication. With Kieran Fisher, my first postdoc in the lab, Elena Maldonado, another PhD student and collaborator. So Andre Kirzak is a computational biologist who was at Surrey. Nick Plant was a toxicologist, and Nick and his colleagues helped me with liver cells in the beginning when I was trying to come to grips with them. But we did a series of computational experiments working on multi-scale modeling of lipogenesis. In particular, we targeted glucose and fructose signaling because of debates in the field that I'm going to mention in a little bit, as well as PPAR alpha signaling, which is essential and dysregulated in lipogenesis. Now, because I'm nervous about my time, I'll skip all the details, but you can ask Kieran about it later. In terms of working translationally, which had always been a goal of mine, particularly inspired by the NIH and working with physician scientists, and also inspired by Sue's research, we started looking at vitamin D in pediatric liver disease. And just to point out, so vitamin D is arguably both a nutrient and a hormone. We can synthesize vitamin D from precursors from sunlight, but we also get it in the diet. Vitamin D has a specific receptor within the cell, and that receptor, in combination with other nuclear receptors, regulates downstream gene expression. Now, vitamin D does a lot of things, and it's very well known for its effects on bone homeostasis. But I think what was of interest to me at the time is its roles in terms of being anti-proliferatory, anti-inflammatory, and anti-fibrotic. And that had been well worked out in some colon cancers, but I was interested in examining it in steatotic liver disease. And pictured here is our clinical collaborator who was then at King's. So Pippa's work basically showed that vitamin D intakes were much lower in children living with liver disease as opposed to obese counterparts. Throughout the year, even when we think vitamin D levels should be at their highest in the UK, children living with the disease had less optimal intakes. And also that vitamin D status related to their liver disease. Simultaneously, at Surrey, we managed to recruit Dr. Barbara Fielding from Oxford, which was a huge coup. And Barbara is an expert in stable isotopes, which is a way of us really measuring metabolism in humans. And as I mentioned, in terms of my interest in glucose and fructose, at the time that was driven by this paper, which was an association paper, but that suggested that fructose consumption was what was driving fibrosis severity. So initially, Barbara and I's first foray into working together was to approach that topic, combining our understanding of both the molecular biology and the metabolism, because it didn't kind of intuitively make sense. Fructose is something that we used to give diabetics. But as we continued to work together, the policy environment around us shifted and changed. And in 2015, both the WHO and the Scientific Advisory Committee for Nutrition in the UK had really pointed out that the vast majority of us are consuming too much dietary sugar. And in 2015, PHE, as sort of the government body, suggested that we needed to reduce sugar in the foods in our diet, and we should start with the foods that were most highly consumed by children that had the highest amount of sugar. So on that list was lots of things that you might expect: cake, candy, those sorts of things. But yogurt was also on the list. And this is an example of a completely interest-led sort of, viral phenomenon that you would never predict. But I looked at Barbara and I said, how much sugar is in yogurt? And I did so because when Claire was weaning, these little pots here, she absolutely adored them. And I commented on the time and how much she loved this yogurt. And unbeknownst to me, they were little pots of sugar. So Barbara and I set about doing this systematically, looking at the sugar in yogurts. And essentially what we found is that some of the highest amounts were found in both children's yogurts and organic yogurts, actually. So it turned out this was really of significant interest to the world. It was all very exciting there for a little bit, and our story carried on a lot of news media around the world. And just to point out, I did a follow-up paper a few years ago, and the UK did challenge the food industry to reformulate their policy. And these little pots of sugar those have been reduced by 3 grams of sugar per pot since this work. And these data show that across children's flavored and organic yogurts, the amount of added sugar has come down. Policy can work as a lever. Although I trained as a molecular biologist, what this sugar story did for me, kind of unwittingly, is put me in the middle of this huge debate in the media. So some of my scientific interests have been driven by the science, by policy, but also what's in the media. And nutrition is a very polarizing subject. And this debate about sugar versus fat, are we ignoring the science on low carb diets, has informed my attempts to try to understand what is the best dietary intervention if we're going to make a difference to cardiometabolic disease. And we started reviewing this systematically when there weren't very many studies in the field. And this was led by Dr. Sue Keneally, who's a bariatric physician. And then during lockdown, with a great collaborator in Finland and Leanne Hudson from Oxford, we did a really deep dive where we looked at all of the cross-sectional studies, all of the randomized control trials, all of the stable isotope studies. But it's a question that still plagues me. You know, are we ignoring the evidence? And the difficulty in steatotic liver disease and other cardiometabolic diseases is that clinical trials in nutrition are very difficult to do. If you put people on a Mediterranean diet and you're giving them acres of olive oil and acres of nuts, they know. It's not like taking placebo. And then the issue in terms of looking at what diets work in terms of steatotic liver disease is that it's still quite a new field. So more studies have been published in the last several years, and with 2 fellows, Aslihan and Xiaomian, we've really relooked at the data again. So this graph suggests that lower carbohydrate diets in green in comparison to their comparator, and their comparator could be low fat, it could be Mediterranean, it's a mixed bag what that comparator is, will result in a greater reduction of liver fat. We've chosen to use a different mathematical metric to look at this because one of the difficulties in looking at the data in these studies is that liver fat baseline data is often very skewed. So you can have a patient with 10% liver fat average in one study and 50% in another. So there's a process called grading of recommendations, assessment, development, and evaluation that's used in medicine. Our effect size estimate is with very low certainty, and that's driven by heterogeneity in the results and variability in the results. And I want to point out that the studies that are down here that have used very low carbohydrate interventions, they've only gone to 12 weeks. So the studies that have gone longer are kind of sitting here. And then what you can see from the low fat, the higher carbohydrate interventions is that these will reduce liver fat as well. So the story is not closed, but I'll try to wrap it up at the end in terms of what I think we should be doing. So in 2016, I was recruited to the University of Leeds. This was a big change. I was, was home for many years, and really moved into sort of leadership roles. And my first task was building a laboratory from scratch. Dr. Joe Sire played a role in that. This is Joe when we had the cabinetry, but we started with a lab that was completely empty, as well as mammalian cell cultures. James Thorne was my first office mate and then became my lab partner for the last 8+ years. Juan was my first PhD student. And this is actually a picture of Juan. She was the last person out, and we were taking these photos, social distancing, during COVID, which was a really kind of traumatic time. So my first 3 PhD students, Xuan, Xiaomian, Xiaoying, all lived and battled through COVID, but together and with the Thorn Group, I think we embraced and achieved the diversity of scientific teams that Bob Cousins first inspired me to. So Leeds allowed me to stretch my wings a little bit in terms of leadership. At Surrey, the focus had been kind of entirely on the lab. And for a while, I was a research group leader. Made a lot of introverted scientists very nervous by doing speed dating to kind of share research interests. We chaired and organized a 2018 Nutrition Society meeting. And this is a picture from lockdown, when in lieu of being able to reward members of the group by any other means, we had an online NSE Oscars award ceremony. So again, I don't think my introverted colleagues really knew what I was doing. I stopped being research group leader only because I accepted a faculty leadership role. So for 3 years, I was head of the graduate school, and I just, you know, I'm trying to be timely about my talk, but just to say how important PGRs are to me in their career development. And my proudest accomplishment from that time was battling a lot of logistical and organizational challenges, and my colleagues to have an alumni careers day. And specifically what I wanted to do was bring back alumni, particularly those that had graduated during COVID, the people who were stuck in their basements, the people who had been really down in the dumps, and to share their journeys and also their careers. And it was a huge success. And I'm really excited to say that I've taken on a PGR role here in Liverpool, and I look forward to continuing to try to help PGRs because it's a difficult career stage. The other way I got to expand at the University of Leeds was through really wonderful colleagues, and managed to be involved in work that addresses population health equities at a more global level. Some of this was through my good colleague, Mike Zulianak. Mike was my first hire at the University of Leeds, and he's now associate professor in Vancouver. And first with Dr. Harriet Fuller, and then with Dr. Nimi Bitubo. Harriet's work focused on gestational diabetes, which is much more common in South Asian women as opposed to white women. And Nimi is a physician scientist from Nigeria, and his PhD was informed by what he could see in the clinic, which was lots of people living with hypertension and not really understanding the dietary causes of that. And Nimi's work has established tools to try to foster patient-clinician communication and dietary information. And with Professor Yunyun Gong, I got to move back to micronutrients, and in GCRF-funded work, we were tackling the double burden of malnutrition in multiple countries, China and Vietnam. And I want to credit here the work of Zhang Yan and also Chloe, who were incredibly hardworking, and many papers have come to this and have allowed me to go back to thinking about zinc and iron deficiency as well as vitamin A deficiency. And an absolute highlight for me was getting to visit China for the end of grant workshop last year. It was really amazing. So I'm only showing the papers that came from the micronutrient side of work, and I know Union has lots more in progress with some of the more eating behavior side of things. In my own group, we continued to work on vitamin D. This was led first by Juan and then by Xiaomian. And when Pippa had started the work looking in the children, we were also interested in particular microRNAs regulated by vitamin D. And this is because, at least in colon cancer, there's an established route by which microRNAs are how vitamin D mediates its antifibrotic effects. So I'm not going to give you all the details, but I just want to let everybody know. So mRNAs won the Nobel Prize this year, so that they're back in vogue, I hope. But the actual receptor for vitamin D is regulated by 2 microRNAs, and one of the most important immune proteins, the signal of cytokine signaling, is impacted by a vitamin D regulated microRNA. So with these guys, we were asking the question, what microRNAs are dysregulated in steatotic liver disease? Which ones are dysregulated by vitamin D? Which of those are in common? This work is still ongoing, and it is some of the work that I'm really excited to do in the new facilities here in Liverpool. I continued to work on nutrition interventions, and in particular, one of the things that interested Sue and I in that first paper is sort of the synergistic effect of nutrition and exercise. So if you're comparing nutrition versus exercise, if you change your diet, you'll lose more weight. With exercise, you usually don't get as much weight loss, but you can profoundly affect metabolism. And so with these collaborators, Professor Suzanne Norris, hepatologist in Ireland, my good colleague, Dr. Chiara Wright, we set about doing an intervention to examine that. Again, I won't give you all of the details because I have no idea how long I'm going on for. But suffice to say, there are differential effects. These appear to be mediated by a particular T cell population known as mate cells. And what we see are differential effects in terms of diet versus exercise. And it appeared in a small study that exercise will affect fibrosis, whereas the dietary intervention will affect steatosis more impactfully. This was a small study. Trying to get paired biopsies after a 12-week intervention is pretty difficult in terms of both getting ethics and reviewing, bringing in participants and volunteering to give 2 biopsies. But the exercise piece and its impacts on fibrosis have since been confirmed by another group. And then the last piece of thing that I want to mention in terms of the Leeds period, and this is very much thanks to Doug Kell, who at some point in the mid-teens said to me, Bernadette, you should look at ergothymine. And with Zhao Ying, and again, James Thorne, and also Paul Taylor, who is a proper chemist, we've been looking at ergothymine. So if you've never heard of ergothymine before, this is a naturally occurring amino acid that we find in our foods. Low amounts in many foods, but very high amounts in mushrooms and fermented foods in particular. And there's been very little work done on it in humans, but I suppose what really caught my attention is a long Swedish study that looked at metabolic markers associated with mortality and cardiometabolic disease, and essentially high levels of ergothymine associated with much lower odds of mortality and cardiovascular disease. How it works at a cellular level appears to be through its own direct antioxidant functions, and also through activating what's known as the master antioxidant response regulator, NRF2. And again, this kind of relates to why Liverpool was able to persuade me to come. And this is a picture of myself and Zhao Ying in China in December, when I had my first experience of a mushroom hot pot. And this was really exciting because when they bring it to the table and they put it in the pot, they set a timer and you're not allowed to touch it. And then they come back with a little container that they're taking a sample of that they're going to hold for 3 days in case something happens to you. So it was all very exciting. It was also delicious, and it turned out not to kill me. Which kind of brings me to my Live Uni era. And if I hadn't already accepted the job, I might have done with this campaign that Leeds ran in advance of Taylor Swift coming to Liverpool last year. And apart from the Department of Biochemistry, so I just can't say how honored I am to be here. I had met Rob Bain at the proteomics meetings years ago. I had invited Claire to come speak to Leeds. I knew the work of this department. But when I came to test the waters as a sabbatical, probably what really sealed the deal is the Human Liver Research Facility. And Chris and Ian and Rowena and Laura and Amy have just welcomed us with open arms. And what this facility has is some of what I wanted to establish in Leeds and really couldn't, although I have a colleague who's doing so now for logistical reasons. But they have established human models, 3D spheroid cultures, precision tissue slices of liver samples. So this really allows us to look at really as close as to a human model as possible. And they've been incredibly welcoming to myself and Xiao Mian. And Amy and I recruited our first PhD student and got that good news this week, which is really exciting. I do want to continue doing both molecular and translational work. At the moment, what that looks like is continuing to look at vitamin D regulated microRNAs, but now in physiological relevant models. With Aslihan and Abby Merrill, we have already received our NHS ethics approval, but we're putting in an amendment, and the study is about to start looking at dietary ergothioneine in preeclampsia. And Song Dai, who we got confirmation on his funding this week, is going to look at ergothioneine and mitochondrial dynamics. It's busy. It's been a busy time since I got here. So there are bids in, one in particular with Rick Dunn that would look at microbiome that I have all my fingers crossed for. Abby, Doug, and Louise and I have put in a grant in terms of looking at ergothioneine, doing an intervention trial with ergothioneine. And with Mike and Nimi, multiple bids have gone in, both from Canada and from the UK, to try to continue on some of Mike's work and hopefully get Nimi here. And bids planned. We're still trying to get some pilot data, but really keen to tap into Richard's proteomics expertise, Rick's metabolomics expertise, Ian on NRF2, and with Paul Taylor, molecular evolution related to ergothioneine has really pointed us to some novel transporters. Okay. Hopefully I have not been talking as long as that timer says. But I'm just going to wrap it up by trying to summarize what I know to be true, I think, or what my truth is. The first drug for steatosis was approved last year. It's a thyroid hormone receptor agonist. It's not going to be the be-all, end-all. GLP-1s, GLP-1 receptor agonists are likely to get approval for steatotic liver disease in America quite soon. There have been positive results from some of those trials. But the field is still very open. If you compare how many drugs there are for diabetes, we're going to need a lot more drugs than are what are currently on the market. And so for the last 15 years and still, nutrition and lifestyle is the cornerstone of clinical recommendations. And when you look at sort of how the disease progresses, originally it was thought you go from normal to having steatosis to having steatosis and fibrosis, and then you had severe fibrosis, and then you eventually either got cancer or needed a liver transplant. There's loads of questions over this natural history because you have to get biopsies from people, and that's not easy to do. So one key question is whether or not there's a subgroup of people who are going to develop more severe disease. A really scary question with quite a bit of evidence to suggest yes, is that liver cancer, so living with obesity is really associated with much higher risk for liver cancer. So can you end up with HCC straight from steatosis? So the things we know from the studies that have been done, these natural history studies, is that what predicts progression is weight gain, saturated fat, sugar. There are question marks over vitamin D and choline deficiency. This is really because of the nature of the field. Things that are associated with regression is weight loss, exercise. The Mediterranean diet is used a lot, and I like to speak about the principles of the Mediterranean diet rather than it in particular. There have been trials done investigating vitamin E and omega-3, and then there's a host of other polyphenols and food biomarkers that we know play a role in the disease. And the difficulty with these food bioactives is it's not any one thing, right? So it's very difficult to do an intervention study with one nutrient and expect it to magically fix a disease that's taken 10, 15 years to develop. But undoubtedly, all of these micronutrients do play a role. Coffee is good for the liver if you can afford it. It's getting more and more expensive. So because I knew I would be running out of time, this is a summary. If you want all the fine grains of the evidence behind it, I'll give you that later. If I could wave a magic wand, I am so tired of hearing low carb versus low fat. I really wish we could shift the dialogue to talking about the quality. Their fats and carbohydrates come in all sorts of different flavors. Based on our most recent review, it does look like lower carbohydrate will help reduce liver fat. And we know that that's true in type 2 diabetes. So if you're living with diabetes and you switch to a lower carbohydrate diet, you have the chance of being able to put your diabetes in remission. The difficulty for liver disease is that very low carbohydrate diets are dangerous for the liver. They're most often not sustainable. So I think what is probably prudent is trying to message healthier carbohydrates. That's looking like eliminating free and added sugars, especially as sugary beverages or alcohol, and understanding that carbohydrates are not bad. So we talk a lot about the gut microbiome, but the gut microbiome will not thrive without fiber, without whole grains. And certainly in the UK, we undereat vegetables and legumes a huge amount. The debate around saturated fat is upsetting. Many committees all over the world in diverse countries have scrutinized the evidence, and the work that I did with Hanela and with Leanne, it's unequivocal. If you overfeed people and you compare overfeeding them saturated fat, unsaturated fat, and sugar, saturated fat is the worst in terms of inducing insulin resistance. So it is just a myth that saturated fat is good for you. Can it be eaten in moderation? Sure. Saturated fat in dairy might be better for you than saturated fat in a sausage. So there are nuances, and I suppose that's my big message. There's nuances to all of the debates and the polarized debates that drive algorithms on social media don't lend well to nuanced messages. If you want to increase the good in your diet, nuts and seeds, virtually nobody in the UK eats them. We should all be eating a portion a day. They're high in fiber, high in healthy fats, high in protein, high in polyphenols, high in micronutrients, and we don't eat them. Olive oil, I put rapeseed oil on because I can't help myself but mention the seed oil debate is also equally annoying. Rapeseed oil, cold pressed from the UK, is a fifth of the cost of olive oil, and it has the same fat profile as olive oil. Some of the issues with seed oil is that industry reuses them and they're put in the worst foods. So they're put in our biscuits and our cakes. And we should be consuming more omega-3 fatty acids. So the principles of the diet, and this is where I want to come back to systems biology. So when we talk about plants, it's not one nutrient. And when we talk about polyphenols, which are all in very tiny amounts, we have to think about these these micronutrients having synergistic properties. So it is the totality of what we're consuming. And the more whole plants you can eat, the better off you're going to be. And it's a whole different talk on exercise, but don't forget exercise. And this doesn't need to be the gym. This could just be walking. It could just be doing a little bit more than you're doing now. So I'm going to wrap up slowly with my philosophy, my opinion, my belief that cardiometabolic diseases are an emergent phenotype that come from genetic and environmental factors. Pathogenesis of steatotic liver disease is complex, but lifestyle, it isn't a modifiable environmental factors. The response to foods, diets for any one individual is going to be different. It is emergent, it's synergistic. And again, a completely separate lecture that I'd love to give is that our current food and social media environment is absolutely prohibitive. It is incredibly difficult to eat well in the environment we find ourselves here in 2025. And as I mentioned, I've made these nutrition recommendations, but 1 in 4 children in this country are food insecure. They don't have these choices. So the longer I've stayed in obesity, the more convinced I've been that systemic issues, socioeconomic inequalities need to be the focus rather than tut-tutting over somebody's diet. So in closing, about my circuitous journey with a significant lag time for the enzyme biologists in terms of my publications, I want to speak about my inspiration for my metaphor. And that's a poem that likely many of my UK and European colleagues will not have ever heard of, because it was by an existentialist American poet, Robert Frost. And, you know, the existentialists focused on the individual and, you know, what to do in a universe that had no meaning. So I'm not going to read the poem, but just to say the protagonist is walking and finds themselves at a fork in the road and finds themselves quite indecisive. And in the third stanza acknowledges that once you take one particular path, I doubted if I should ever come back again, and I'm reminded of epigenetic decisions here. And the very last stanza really speaks to me. 2 roads diverged in a wood, and I took the one less traveled by, and that has made all the difference. So just to say a huge thank you to my mentors, students, collaborators, colleagues, friends old and new. It makes a lot of sense to me that my journey has ended here in Liverpool, where even if you're a blue, you'll never walk alone. And me and my family are thrilled to be here. And thank you for listening.
That's your kids.
I've never seen such a good piece of poetry. And you know which way your bread's buttered because you boiled it implemented, so well done. So I have 4 jobs before we'll disappear upstairs for our drinkies. The first actually is to thank all the people that have come here today. We are going to let there be a few questions, but I also want to really push home, this is nothing without you coming and attending and being with us. And we know, obviously, Bernadette's close family, Nick, Claire, and Xavier. Thank you, Xavier. I can never quite get it. You guys are the ones that keep Bernadette going, and I'm sure vice versa. But also, Bernadette really has asked me to thank you. You come from all around the country, and I know it's always funny when you're online, but we know people who come from London, Sheffield, Leeds, but also I wanted to welcome everyone in the USA, Canada, and hopefully Africa as well. I think that really shows just the worldwide influence that you've had, Bernadette, and that others think enough of you to attend something like this. So many thanks to everyone for coming. Give yourselves a round of applause. Okay, so my second job is to ask Bernadette if she'd be happy to take a couple of questions if there are any. And you can stay sat down if you wish, please, because it's exhausting giving a lecture of that length. Thank you so much again. Do we have any questions for our speaker, our inaugural lecturer? And there's one at the back there straight away. Thank you.
So Tim Cross,
Sorry, Tim, I can't see you clearly.
That's probably better for everybody. I'm Tim, I'm a liver.
Hi, Tim.
I really enjoyed the talk. Thank you so much. That was brilliant. I just wondered if you'd comment on how we've moved from, with the increase in fatty liver disease over the last 20 years, how we've moved from, say, an agricultural to an industrial diet. And I wonder what your thoughts, because on ultra-processed food and processed food, how that's changed.
I see question.
So, no, it's all about this primary.
Yeah.
So, I'm interested to hear your thoughts.
Yeah. Hopefully I won't be too long-winded. Again, I could give an hour lecture on this topic. So ultra-processed foods is a new term and a new classification. And before the term ultra-processed foods, we've used terms like the Western diet or the junk food diet. And the term ultra-processed foods comes from a particular system developed by a Brazilian scientist. And again, like many other things in nutrition, there's nuance here. And I think that the reason I love systems biology is because I firmly believe that two things can be true at the same time. So on the one hand, food processing is why we're all alive. Food safety is what the vast majority of food manufacturers worry about. We have the safest, arguably most abundant in terms of nutrients, food supply that we've ever had. So it's how we feed the world. Equally, the food industry has a lot to answer for in terms of many of the ultra-processed foods, what we used to call junk foods or Western, are in fact really high in fat, really high in sugar, really high in salt, also very palatable and yummy and easy to eat. They're mushy and they digest quickly. So the debate in the field, if there are folks here who aren't tuned in, on the one hand, there's a group of people who are convinced that ultra-processed foods are killing us and that the food industry is the devil. And on the other hand is a group of scientists that are saying, is it the processing per se, or is it the rubbish nutrient profile, or is it how hyper palatable the food is? And the real difficulty with the NOVA labeling system is that it's not perfect. So it lumps into ultra-processed foods things like bread. What is a healthy food? It's contextual, right? If a child is not going to get any other fruit and vegetables and they have a glass of orange juice, have at it, right? If you have not eaten and your only choice in terms of food or no food is chips and sausage or chips and fish fingers, then any food is better than no food. So the definition is problematic. I think the issues in terms of our industrial food environment is more than the food. We know the food that's not good for us, right? The issue is our high street. The issue is bubble tea. The issue is all the dessert restaurant places that have popped up.
Oh, great.
Oh God. You planned that, didn't you?
No, it's my vegan sausage roll, which I have every Thursday.
So Pat has started keeping fruit in his office because of me. So great question. I think the food industry has a lot to answer for, but again, going back to that time in Washington, D.C., we cannot change the system by throwing the baby out with the bathwater. And at least, you know, if we think about, so, you know, the people who sell us food, Sainsbury's, Tesco, and all of that, I've worked closely with nutritionists at quite a high level there. They are really working to make change, but they're working against capitalism and people's likes. So people don't buy the vegan sausages, they buy the regular ones, or whatever. And it's worth pointing out that we do need to eat more sustainably, and many of vegetarian and vegan products are by nature ultra processed, as is soy milk, as is, you know, formula, which is life-giving. So I think it's difficult. And what I don't appreciate, and I'm happy to say this for the record, are people who have vested interest slandering nutritional scientists. So I think if you're selling books, I think if you're selling a podcast, I think if you're selling a gut microbe shot or 30 plant things in a little pot, you should declare that when you're trashing nutritional scientists that are really doing their best. And one of the peculiar things about nutrition is that, so every time I go to a hepatology conference, every talk starts with a PowerPoint slide in tiny font of all of the money that has pharmaceutical funding medical research. That's how it gets done. But if a nutritional scientist or a food scientist takes any money from industry to make the food supply better, we're demonized. And there have been some hit pieces in the BMJ that have been disproven and have been argued. But in the meantime, they're out there in the Daily Mail. So I have a watch this space on ultra processed food. I think, again, the reason they're not good for you is synergistic. I don't think it's just the nutrients or just the hyperpalatability or just the marketing, which Emma Boyland studies here. I think it's all of those things.
Douglas.
Fabulous talk about that. I especially liked the what I can consider the two-word summary, which is go nuts.
Go nuts. You heard it from Doug first. Yeah, if you want one small way that you can make your diet better, go buy raw nuts and seeds and consume a small handful daily. If you're trying to get up your protein, pop it on your full-fat yogurt in the morning. And mushrooms. Oh yeah, no, Doug eats a lot more mushrooms than I do. Yeah.
So Richard had a question.
Yes, thanks for the lecture. As expected. So you just mentioned sustainable food sourcing. So looking forward, we know we need to change how we source food and especially the protein intake. So what's your vision going forward? How are we going to settle this and how are we going to keep that healthy apart from the palatability?
Yeah, so it's not exactly my area. And interestingly, one of my absolute scientific heroes, Professor Anwesha Sarkar at the University of Leeds, has just led a bid for a program of work over the next 5 years developing alternative protein sources. So this is a huge consortium of both universities and industry partners. And that, you know, I don't know all of the details, although I did read the bid. That is looking at all angles, from insect proteins to Doug's sort of biosynthetic methods of fermentation and finding other ways of getting protein. The processing piece is a difficult dance to dance, you know, within that piece. They will be ultra processed foods. And certainly if we look to Asia, we can see that the ultra processed protein sources already exist. I have to say, personally, I fall in the camp that I really, you know, I would much rather have a portobello mushroom burger than a meat alternative type burger. But I think we need all options. But it's not necessarily something I'm going to work on myself. And I think the human studies need to be done, but they very rarely are in nutrition. So, or not in nutrition, but in our food supply. Industry manufacturers make new food products all the time, and they either sell or they don't sell. So there was these sausages that had put in legumes, so they'd reduced the meat down by 25%. I loved them. My kids, I couldn't get them into them at all. And like 2 months later, they were off the shelves. So if they don't sell, it's gone. And we very rarely, so at the moment, inulin and stevia, these are sweeteners that are natural, that we're adding to our foods, and nobody's done trials to see what effects those have. So if that grant comes off with Rick, that's one of the things that we want to look at, is specifically how these nutrients affect the gut microbiome and how that influences things. There just isn't enough money. And to do the trials for how long? You know, how long would you need to eat insect protein before you learned if it was good or bad for you? But people do eat insects all around the world.
Okay, so we will take one more question from the soror.
One receptor agonist. Now, this is very popular these days. The injection that we're giving.
Yeah.
And the GLP-1 and GLP are government. They have to bind the receptor to get the effect, and that is why they go for agonist of the receptor. GLP-1 receptor is all over the body. In practice, it uses insulin secretion, and it is in the liver.
Yeah.
And do you think that that would be because in heart and kidney seem to be related conditions of disease? What about the liver disease?
Yeah, so a great question. So this is from a legend in the field of nutrient sensing, for those of you online who might not be able to hear, and she's asking about GLP-1 receptor agonists in liver disease. So the first thing to say is, I think this is the most exciting class of drugs that have come about since statins. So far, all the trials, so the Essence trial is specifically looking at liver outcomes in response to GLP-1 receptor agonists. I can't remember which one. And those trial results were published presented in November, interim 1-year mark, and they were very positive. So everybody in the hepatology community is anticipating them being approved by FDA for use in America. Like I said, I think these drugs are really exciting. Diet and exercise will only induce a very small amount of weight loss. And that can be really disheartening for somebody who's living with a lot of obesity. The problem is how they're prescribed, and they're now very much available through online pharmacies, and there's no nutrition or behavioral change support. So the foundation of the Canadian guidelines on obesity, which are really excellent, speak about medical nutrition therapy needing nutrition, exercise, behavioral therapy, as well as either pharmacological therapy and bariatric surgery. These are all the ways we can treat and support a patient living with obesity. So all of the data presented in the last month or so at the big obesity meetings have shown that as soon as people go off these drugs, they gain weight back, and they gain it back faster. So there is a piece of data that shows in a very managed behavioral intervention post people stopping, supported by diet and lifestyle change, that people didn't gain weight. So it is possible, and if anybody needs a recommendation on a really fantastic physician, I can give you one. It is possible, but I'm worried at the moment. 13% of Americans have been on one. We don't know the long-term effects. They don't induce weight loss in everybody. All of Novo's, paraphernalia says you need to make diet and lifestyle changes alongside it. Some people find that really easy to do. The dual agonists, GLP-1 and GIP definitely influences the liver. And I guess I'm just going to close by saying what's really important about this class of drugs in terms of stigmatization of obesity is it proves what I been teaching for many years, which is that fundamentally obesity is a neurological disorder, and many people, maybe not all, are hungry. They are hungry. And so the number one thing that these drugs do is induce satiety. So a lot of people, while they're on the drugs, are able to make changes that they weren't able to make before. The question of their long-term effects, we have studies going out to 96 weeks, but we don't have much longer than that. There's an accessibility issue. So here in this country, they've just made it possible to get these drugs from your GP if you have a BMI of 40, but that leaves a lot of people quite overweight that don't have access to them. So I think it's an interesting space, and I would recommend them to a friend who is struggling at the moment. So far, the safety data looks great. There's data suggesting they reduce mortality and long-term endpoints as well. The liver data is still forthcoming.
Okay, fantastic. I don't think I've ever heard such eloquent answers to questions.
Oh, Pat, you're really trying to butter me up now.
We'll talk about the teaching load later.
Okay, here it comes. Here it comes.
No, seriously, before we do the claps and the presentation, I'd just like to say, please join us for drinks upstairs. That's right, Jackie, isn't it? Good, because I know you haven't started with me yet, Jackie. I didn't mean it like that. But so we're gonna have a drinks reception upstairs. Please come and join us to celebrate Bernadette's inaugural. It's also a tradition that for all biochemistry inaugurals, we then go to the Philharmonic, where I have to bear the first drink. So honestly, I'd probably go home tonight early because the weather's looking a bit rough. It's probably going to rain. But we would love to see you obviously in the Philharmonic as well. And so that's my three first points. The final point, of course, is to thank Bernadette for everything. When we were trying to recruit you, I think it was very clear that you had lots of great ideas, you had an incredible track record. But I think what really got me most was that when you're around, it's a bit like having a ray of sunshine.
Aww.
So we only had to choose the flowers you learned about. We went for sunflowers.
Oh, my favourites.
Are they your favourite?
They are my favourites.
That's right.
Yes, thank everybody online. I haven't been able to keep up with the chat, but I love you all, and I look forward to catching up with you very soon. Thank you so much. Sue, I can see you. Thank you very much. Means the world.
Awesome.
Sorry for going on the bush.
Well done.